My name is Thomas Hatfield in Bio 181, and I will be doing my report on Christos Katsanos. Christos Katsanos is an exercise physiologist, and he studies metabolic responses in human obesity. His current focus is on metabolic abnormalities developed in skeletal muscle secondary to obesity. And he also specializes in nutrition, obesity, diabetes, exercise, kinesiology, and metabolism. He got his PhD at Florida State University, his MS in University of Louisville, and he also studied over at in Greece. So let's dive a little deeper into the experiment. Um, the exact experiment is about metabolic responses in human obesity. And a little background: obesity leads to many other health conditions. Obesity is affected by an individual's metabolism, and metabolic syndrome can occur, and that means insulin resistance, B cell failure, impaired glucose tolerance, and type 2 diabetes and other diseases. So, this specific experiment was titled Prolonged Exposure of Primary Human Muscle Cells to Plasma Fatty Acids Associated with Obese Phenotype Induces Persistent Suppression of Muscle Mitochondrial ATP Synthase B Subunit. There are lots of authors on this experiment, and it was on the website of U.S. National Library of Medicine. So just a little introduction. The increasing prevalence of obesity is a major health concern due to its associated physiological and metabolic consequences. This is linked to cardiovascular disease and type 2 diabetes, as I already said. And in the experiment, they evaluated these same responses in a primary cell culture model and tested the specific hypothesis that circulating non-esterified fatty acids, which I'll be referring to as NEFA, and obesity play a role in the responses observed in humans. So in this experiment, they hypothesized that the plasma and NEFA profile associated with obesity reduces total protein synthesis in a muscle, and at the single protein level suppresses the translation of efficiency of BF1 ATPase, and the latter effect is mediated by miRNAs. So the experiment was fairly complex, but just to be brief, they compared lean and obese patients and had them fast for 10 hours. They took blood and muscle samples that were processed, extraction of non-esterified fatty acids, quantification of mRNA, miRNA, and protein, concurrent glucose uptake and protein synthesis, inhibition of MIR 127-5P experiments and exosome isolation, and DNA methylation. They took a lot of numbers and were able to compare them, which leads us to a small part of the results. Obviously, in these images, we can see the lean and obese subjects being compared, and the NEFA, that was the levels in each one, and how that affected their glucose and their protein. The experiment describes these images as concurrent assessment of glucose uptake and nascent protein synthesis in primary human myotubes reveals independent regulatory mechanisms. So finally, in the journal, it says, we provided in-depth mechanistic insights into how obesity perturbates skeletal muscle protein metabolism as it relates to BF1 ATPase, and advancing previous findings on mitochondrial protein metabolism in obesity. Our data suggests that this response of the muscle of obese individuals is linked specifically to the plasma NEFA profile associated with obese phenotype. Furthermore, our findings implicate certain epigenetic mechanisms involving miRNA regulation is sustaining the impaired translation of muscle BF1 ATPase involved in energy metabolism. So they go on to make a couple other claims that there is a large variability in protein expression in muscles of obese compared to lean individuals at the single protein level in general and mitochondrial level in particular. And they also say that the findings suggest that a key element contributing to suppressed BF1 ATPase metabolism in muscle is chronic exposure to lipids associated with the obese phenotype. This is obviously very important, and any information we can get on our bodies and the health of others, especially here in America where obesity is such a problem, is something very applicable today. So that's my presentation. Thank you for listening.